Hello and welcome to lecture 19 of our course on stochastic modeling of biological processes. In our previous lecture, I showed how active transport can be incorporated into stochastic reaction diffusion algorithms, including both individual-based and compartment-based models, which were based on position jump processes. In this lecture, we will discuss reaction-diffusion-advection models, which are based on velocity jump processes. In section 7.3, such models are applied to modeling behavior of bacteria, where we will discuss how we can derive population-level equations from individual-based models. Another example of agent-based modeling is presented in section 7.4, where we will discuss collective behavior of locusts. In section 4.3, we presented velocity jump processes, modeling diffusion in a certain limit. The simplest one-dimensional velocity jump process was formulated as follows. A particle moves along the x-axis at a constant speed s and reverses its direction at a random instance of time. The probability of turning in the infinitesimally small time interval dt plus dt is equal to the turning frequency lambda times dt. Choosing the turning frequency as s squared divided by 2d, we showed that for sufficiently large times, this process models diffusion with diffusion constant d. To add signal-dependent advection to this velocity jump process, we make the turning frequency dependent on the concentration of signaling chemical in the environment, signal Sx, using this formula, where b is a constant. If b is equal to 0, then we have the original unbiased velocity jump process. If b is not equal to 0, then the jump rate to the right will be different from the jump rate to the left. This is achieved by this plus-minus sign. I will use one of these signs for jumps to the right and the other sign for the jumps to the left. The choice of the signs will depend whether the signaling chemical is an attractant or a repellent. In section 4.3, we analyze this velocity jump process by writing evolution equations for densities p plus and p minus, which are densities of particles that are moving to the right and to the left respectively. This system of partial differential equations is still valid even for my modified turning frequency lambda. So let us substitute formula 723 for lambda. We obtain the following equations, where you can see that I am using the minus sign for the turning frequency of the right moving individuals and the plus sign for the turning frequency of the left moving individuals. In particular, if b is a positive constant, then the signal Sx will be an attractant, and if b is a negative constant, then the signal Sx will be a repellent. To derive this conclusion, think about s, which is an increasing function. Then the derivative of s is positive. If b is itself positive, then this term is positive. So the right-moving particles, which are going in the direction of increasing signal, are less likely to turn than the left-moving particles, which are going in the direction of the decreasing signal. Therefore, this choice of signs indeed models an attracting chemical signal for positive values of parameter b. Next, we can follow the approach presented in section 4.3 to derive the hyperbolic version of the Chemotaxis equation. Considering b is equal to 0, then the red term is missing, and we obtain the telegraph equation, equation 446, which we derived in section 4.3 for the density of particles. The long-time behavior of the telegraph equation is given as a solution of the diffusion equation. That is, we can drop this black term and we are left with the blue terms, giving the diffusion equation with diffusion constant d. You might have noticed that this equation has an extra parameter called lambda 0, which I have not yet defined. This parameter lambda 0 is just equal to s squared divided by 2d. In section 4.3, we considered an application to modeling diffusion of proteins, where we knew their diffusion constant d, and we estimated their speed s, and we used the formula s squared divided by 2d to estimate the corresponding turning frequency lambda. In section 7.3, we are in a different situation. We will be applying velocity jump processes to modeling bacteria. In this case, lambda 0 is a measurable quantity. It is the turning frequency in the case when the signal is constant and the red term is missing, so we can estimate it from some experiments. In particular, we could define our velocity jump process in terms of the parameters lambda 0 and b and derive the hyperbolic chemotaxis equation in this form. For long times, we can drop the black term and we arrive at the chemotaxis equation. Starting with a simple velocity jump process with signal-dependent turning rates, we have derived the classic chemotaxis partial differential equation, which has been used in deterministic modeling of chemotaxis during last 60 years. I have written it as two partial differential equations, one for the density of cells and the other one for the density of the external signal. To formulate this system, which is sometimes called the Keller-Segal model or the Patlak-Keller-Segal model, we have to also specify the functional forms of various terms, like the chemotactic sensitivity, which in our previous derivations was a constant, but it could also depend on the signal S. The signal S could also be produced by cells, or consumed by cells, or degrade on its own. Depending on the choices of these functions and parameters, there are many interesting properties of these equations, which are studied by mathematicians. So it is good that we could also derive this equation from an individual-based model. However, we already know that other stochastic diffusion-advection models also lead to the chemotaxis equation at the macroscopic deterministic limit. So an important question remains. Can we derive the chemotaxis equation by considering how an individual bacterium sense and respond to environmental signals? To motivate this question, I have included here some videos of real-time imaging of bacterial behavior from the laboratory of Howard Berg at Harvard, where you can see the velocity jump process in action. Bacteria swim straight with constant speed, and at random instants of time they turn and move in a different direction. These videos show swimming bacterium E. coli, which is the most widely studied prokaryotic model organism. We can summarize these observations that E. coli has effectively two modes of behavior based on counterclockwise and clockwise flagellal rotation. Counterclockwise rotation pushes the cell forward in a straight run, while clockwise rotation of flagella triggers a random tumble that reorients the cell. For E. coli, the duration of both runs and tumbles are exponentially distributed with means of 1 second and 0.1 seconds, respectively, so the tumble is quicker than a run. So we could just assume that it's instant. Therefore, the bacterium is undergoing the velocity jump process. It runs with a constant speed, and at a random instance of time it changes its direction. This velocity jump process is biased according to the environmental signals. Roughly speaking, a bacterium compares its current environment with the environment it visited a second ago. If the concentration of the chemoattractant is increasing along the cellular trajectory, then the bacterium is moving in a good direction, and it is less likely to change it. On the other hand, if the concentration of S is decreasing along its trajectory, then the bacterium is more likely to turn. To compare the quality of the current and previous environments, a bacterium must have a memory. It must remember the quality of the previous environment. Of course, bacteria are very small and do not have a brain like humans. Their memory is constructed in purely biochemical terms. E. coli detects the attractant concentration by receptors on its membrane. The information about the receptor occupancy is then carried by intracellular signaling molecules to flagella motors. The signaling molecules are proteins which are subject to biochemical reactions, forming a so-called signal transduction network. Depending on the abundance of a particular intracellular protein, a bacterium changes its turning frequency. The summary of the main processes which we have to include in our individual-based modeling is included on this slide, starting with the signal detection using the receptors on the cell membrane. The fraction of the occupied receptors is an input of the signal transduction biochemical network, and its output is a protein which changes the behavior of flagellar motors. In mathematical terms, we have to describe each cell not only by its position and velocity, but also by additional internal variables, which evolve according to a system of differential equations. In the lecture notes, there is a discussion of a relatively simple and analytically tractable model of internal dynamics, for which it is possible to derive the Chemotaxis equation for the density of bacteria. Considering more complicated models of internal dynamics, computer simulations will be the method of choice. Such a computer-based individual-based modeling is an example of agent-based modeling, which is used in many different application areas. An individual agent uses a fixed set of rules based on information about the environment or communication with other agents in order to change its state and fulfill its objectives. In the rest of this lecture, I will discuss individual-based modeling of locusts, but individuals could also be a group of robots. In the Mathematical Institute, we have had a small lab where MSc students have done their dissertations on swarm robotics, working with epoch robots. 8 MSc students have written their dissertations working with these robots to date, so I have included a picture of epoch robots here as another example. Typical questions which one asks for such systems are: how do local interactions between components of a system determine the behavior of the whole system? What changes can be made in the individual level model to achieve a desired change in the collective behavior of the system? In the rest of this lecture, I will discuss section 7.4 on collective behavior of locusts. In 2006, Boole and co-authors reported results of their experiments with locusts marching in a ring-shaped arena. A video of their experiments is shown here. They filmed the arena using a camera from the top and recorded a lot of videos, which they further analyzed. Each of these black spots is an individual locust. Since the experiments have been conducted with wingless juveniles, their movement was restricted to the two-dimensional world. They could not fly away. Since the experimentalists built the ring-shaped arena, they further constrained locusts to effectively moving in one spatial dimension. Such an experimental setup is easier to model, so I have chosen it as our illustrative example in this section. Each locust likes to move in the direction of its neighbors, so there are only two possible types of collective behavior. Either the whole group of locusts move clockwise, or the whole group of locusts move anti-clockwise. The experimentalists used groups of locusts with different numbers of individuals and obtained results of the following form. Here you can see the group alignment as a function of time. Similar graphs can be obtained for the average velocity of the locust group, which I use as our main observable in the lecture notes. We clearly see two well-defined states, which correspond to clockwise and anticlockwise rotations of the locust group. And you can see that the locust group was in one of these states for about 2 hours, and then the group switched their direction of movement. As we have already seen in chapter 3, one of the main questions for the models with two favorable states is the average time to switch between these two states, and how it depends on the parameters of the system. For example, on the number n of the locusts in the group. In the lecture notes, I have written a simple individual-based model for such a behavior. N locusts are marching along the unit circle. Each locust is described by its position, xi, which is given as cosine theta I sine theta I. So the position is effectively one-dimensional, parameterized by angle theta I. The derivative of theta I is its angular velocity, denoted omega I. We assume that locusts can only sense other locusts if they are sufficiently close. For each locust, we define the set of its neighbors at time t by this formula, where r is a dimensionless parameter, the interaction radius. Then ui is the average angular velocity of the locusts which are within the distance r from the ith locus. If it's positive, then the majority of its neighbors will go in the anti-clockwise direction. And if it's negative, then the majority of its neighbors will go in the clockwise direction. So we calculate the sign of this local velocity, which is either +1 or -1. And this is the direction which the ith locust aims to follow according to this stochastic differential equation. Considering the group level behavior, we are interested in the average velocity of the whole group, defined by this formula. Our main question is, how does the mean switching time depend on the number of locusts in the group? If the dimensionless interaction radius r is equal to twice the radius of the unit circle, that is, if r is equal to 2, then each locust will interact with all other locusts. So we are interested in the values of r which are less or equal to 2. In experiments, the interaction radius between locusts is 13.9 centimeters. This number is very small comparing to the size of marching bands formed by locusts nymphs, wingless juveniles, that can extend over many kilometers. The experimental ring-shaped arena also has a circumference 2.5 meters, which is larger than the interaction radius. In particular, the dimensionless parameters r should be chosen smaller than 2. One could argue that given these experimental values, the value of parameter r should be fixed. However, in our computer simulation, we can vary it as well. In fact, we can obtain some nice analytical insights into the problem if we initially choose r is equal to. If r is equal to, then the neighborhood of each locust contains all other locusts, and the local velocity average is equal to the global velocity average. Substituting r is equal to, the stochastic differential equation for the angular velocity of the ith locust is given as follows. Illustrative simulations of this model are presented in figure 7.4, where I plot the average velocity as the function of time for groups with 10 and 20 individuals. We observe that the system still switches between the favorable states of the system. It takes more time for the larger group to switch. And we can prove this. We add equations 743 together to obtain the following equation for the sum of angular velocities. Dividing by n, we obtain that the average velocity of locusts at time t plus delta t is equal to the average velocity at time t plus two terms which resemble a stochastic differential equation. We do have a sum of n independent normally distributed random variables here, but the sum of normally distributed random variables is again a normally distributed random variable. So we can indeed write the equation for the average velocity of locusts ut as a stochastic differential equation. In particular, we can now apply all the theory which we developed in Chapter 3 to analyze this problem. Using equation 336, we can immediately calculate formulas for the stationary distribution, which is presented for different numbers of individuals in figure 7.4. We confirm our observation that the switches between the two macroscopic states of the system corresponding to u equal -1 and u equal +1 will less likely occur for larger groups. We can also calculate the average time for the group to change its direction from u is equal -1 to u is equal +1. The resulting formulas can be written in terms of the error function and the imaginary error function. If n is large, then we can approximate them in equation 747 to obtain that the average switching time depends exponentially on n. Both results are plotted in the right panel. The special case r is equal to could be analyzed using the methods from chapter 3. However, as I have already discussed, the case r is less than 2 is much more biologically relevant. I have left the analysis of this case to exercise 710, where you are asked to consider the model 740 to 741 for the case r is less than 2. You are asked to write a computer code which can simulate this model and investigate how the average time taken for the group to change its direction depends on the interaction radius r in the interval 0, 2. Using n is equal 20 locusts, you are asked to plot the stationary distribution of the average velocity given by equation 742 for different values of parameter r. You can compare such results with figure 7.4c, which was computed for r is equal to, and it's plotted here as the red line. You can test the correctness of your computer code by using the analytical result for r is equal to. This is one advantage of having an analytical result, even if it is not for the most biologically relevant case. Decreasing r, you can observe that the two favorable states become less well-defined, which can be further confirmed if you further decrease the value of the parameter r. If r is equal to 0, then you obtain another analytically tractable case, so there is another opportunity to obtain some analytical results to benchmark your computer code. In exercise 710, you are also asked to study the average switching time as a function of r for n is equal 10 and n is equal 20. And again, your computer code should be matching analytical results for r is equal to or r is equal 0. In the case of smaller values of r, there are no well-defined favorable states. So I have estimated here just the average time to go from the average velocity -1 to the average velocity +1. As expected, it is an increasing function of r for sufficiently large values of r when the two favorable states exist. This brings me to the end of lecture 19, where we concluded our discussion of selected topics from chapter 7. We covered section 7.3 on bacterial chemotaxis and section 7.4 on collective behavior of locusts. Please read pages 200 to 210 of the lecture notes before watching the video of our next lecture. In lecture 20, we will discuss selected topics from chapter 8, covering microscopic models of Brownian motion. Thank you very much for listening to lecture 19 of our course on stochastic modeling of biological processes. Bye bye.